Hey everyone, and welcome to this explainer. Today we're going to take something that honestly might seem like a really dense, intimidating biological process and transform it into what it truly is, a thrilling, microscopic roller coaster ride. We're talking about photosynthesis in higher plants, but we're going to look at it a bit differently. We are going to follow the incredible, wild journey of a single electron. If you're studying for an exam and trying to master this massive topic, you're in exactly the right place. We're going to lock in on the exact mechanisms and clear up all those tricky details that usually cause confusion. Okay, let's dive right into this. Here is our roadmap for today. We'll start off with the basic principles, look at some of the historical discoveries, map out the chloroplast geography, ride our electron through the Z-scheme, and finally wrap the whole thing up with chemosynthesis. Section 1, Basic Principles. To really get this, we need to look at photosynthesis, not just as something a plant does, but as a massive energy problem that the plant literally has to solve to survive. To truly grasp this process, we need to define it super precisely. Photosynthesis is an anabolic process. What does that mean? It means it takes really simple molecules, specifically our raw materials, carbon dioxide and water, and synthesizes them into large, complex carbohydrates like glucose. Notice something here. It is not an amphibolic process. It's purely anabolic, completely focused on building things up. It's also a physicochemical reaction because it's using physical light to drive a chemical change. We call it endergonic because it absolutely requires a constant input of energy from sunlight. And lastly, it is a redox process. Why? Well, because water is oxidized, meaning it loses electrons to eventually release oxygen as a byproduct, while carbon dioxide is reduced to form that final carbohydrate. Grasping these four terms is definitely your foundational key for everything else we're going to talk about. Moving straight into Section 2, Historical Discoveries. We've got to ask, how in the world did humans actually crack the code on what plants need to survive in the first place? Let's walk through this chronologically, because it's actually a pretty cool story. Back in 1770, Joseph Priestley performed his famous bell jar experiment with a mouse, a burning candle, and a mint plant. He essentially proved that plants somehow restore the oxygen that animals in open flames deplete. A few years later, in 1779, Jan Ingenhousz took that exact same setup, but tested it in darkness and then in sunlight, proving that light is absolutely essential. He also did this brilliantly elegant experiment with an aquatic plant, showing that tiny little oxygen bubbles only formed around the green parts of the plant and only when it was in the sunlight. Later on, Moll's half-leaf experiment, which used a KOH solution to absorb carbon dioxide, proved that without CO2, photosynthesis simply halts entirely. So there you have it, sunlight, green pigment, and CO2. The ultimate recipe was uncovered. Section 3, Chloroplast Geography. All right, it is time to zoom all the way in, straight down into the mesophyll cells, to set up the exact spatial arena for our electron's big journey. And this brilliantly illustrates the chemical tools the plant actually uses to grab that sunlight. We have our primary and accessory pigments. Chlorophyll A is the absolute chief pigment here. It appears blue-green. Then we've got chlorophyll B, which is yellow-green, xanthophylls, which are yellow, and carotenoids, which range from yellow to yellow-orange. If you look at how these pigments absorb light, chlorophyll A and B absolutely max out their light absorption in the blue and red wavelength regions. But here is the really fascinating part. If you look at the green spectrum, right around 500 to 550 nanometers, there is literally a flat line in absorption. The plant doesn't absorb green light at all. It completely reflects it. And that is exactly why leaves appear green to our eyes. Pretty wild, right? Section 4, the Z-scheme. Okay, here we go, the main event, the non-cyclic transport system where our electron finally starts its wild ride. But wait, before we launch this electron, we have to ask a really basic question. Where does the electron even come from in the first place? Where exactly does the splitting of water, or photolysis, actually happen? Think about it for a second. Got it? It happens on the inner side of the thylakoid membrane. This is absolutely crucial to remember. Water splits on the inside, releasing its electrons to fuel our roller coaster while dumping protons right into the thylakoid lumen. Keep that in the back of your mind. It's going to be incredibly important in just a few minutes. All right, let's track the exact sequence of the Z-scheme. The journey kicks off at photosystem 2, or PS2, which absorbs light optimally at 680 nanometers. The light excites an electron, practically launching it upward to the primary acceptor, pheophytin. From there, it moves to plastiquinone, or PQ. Now, here is a massive tip straight from the source material. PQ is not just an electron carrier, it is a hydrogen or proton carrier. It actually grabs a proton from the outside stroma just to pass the electron along. Next, the electron travels down the gradient to the cytochrome B6F complex, then over to a copper-containing pool called plastocyanin, and finally arrives at photosystem 1. PS1 gets zapped by light at 700 nanometers, re-exciting the electron up to an iron-sulfur protein, then to ferredoxin, and finally to the FNR enzyme. This enzyme takes the electron, grabs a proton from the stroma, and reduces NADP+ into NADPH. Man, what a ride! Now, I really want to spotlight a super important number here, 8. This is known as the quantum requirement. To liberate just one single molecule of oxygen during that initial splitting of water, the plant needs exactly 8 photons of light. That's 4 photons striking photosystem 1 and 4 photons striking photosystem 2 simultaneously. It is a beautifully calibrated microscopic machine, down to the exact photon. But, sometimes the plant needs to take a little detour. Let's draw a quick contrast between our Z-scheme, the non-cyclic pathway, and the cyclic pathway. Non-cyclic happens in the grana, uses both PS1 and PS2, and produces both ATP and NADPH. But cyclic photophosphorylation happens primarily in the stroma lamellae. Why the difference? Well, it's because the stroma lamellae are actually missing photosystem 2, and they completely lack that NADP reductase enzyme. Without those two things, the electron from PS1 just loops back through the cytochrome complex. And because it can't make NADPH, the cyclic detour exists solely to pump out extra ATP. Section 5, Chemiosmosis, the grand finale. How does the chloroplast actually mint this molecular energy currency? So the crucial point here is that for Peter Mitchell's chemiosmosis mechanism to actually function, you absolutely must have 4 specific components in place. You need a membrane to act as a barrier. In this case, that's our thylakoid membrane. You need a proton pump, which, as we just learned, is our good buddy plastoquinone. You need a proton gradient, which is an electrochemical difference between the inside and the outside. And finally, you need the machine that does the actual forging, ATP synthase, which is made up of the CF0 and CF1 particles. Let's see exactly how the plant builds this incredibly intense electrochemical gradient. It basically uses 3 distinct tricks to stack protons, or H+ ions, inside the thylakoid lumen. First, like we discussed earlier, the photolysis of water happens on the inner membrane, directly dumping protons right into the lumen. Second, as plastoquinone passes electrons along, it actively pumps even more protons from the stroma outside into the lumen inside. And third, on the outside stroma, the reduction of NADP+ to NADPH physically consumes protons, draining them from the outside. The end result? The lumen becomes totally packed with protons, while the stroma is heavily depleted. The plant has essentially created a massive, tightly coiled spring of electrochemical energy. And here comes the release. Those protons desperately want to escape the highly concentrated lumen and flow back out into the stroma. But their only exit door is through the CF0 part of ATP synthase, which is an integral transmembrane protein. As the protons violently rush through CF0, it triggers a physical conformational change in the CF1 particle hanging out in the stroma. This peripheral CF1 protein literally acts like a microscopic turbine. The rapid flow of protons makes it spin, and that mechanical energy violently forces ADP and inorganic phosphate together to forge ATP. The electrochemical gradient is successfully broken down, and the plant has minted its energy currency. It's just amazing. We've literally followed a single electron from the splitting of a water molecule all the way down to the spinning turbine of ATP synthase. It is an absolute masterpiece of biological engineering, and it's happening by the trillions inside every single green leaf you see. Which leaves us with this final, kind of mind-blowing thought. If we can fully map and perfectly understand the chloroplast's microscopic sun-powered ATP engine, could humanity eventually build artificial leaves to solve our own global energy crisis? The answers to our biggest future problems might just be locked inside these tiny, green, microscopic machines. Keep studying, keep asking those big questions, and I'll see you in the next explainer.